start that. Okay, so hello everyone. Thank you for coming. Today I'm going to talk to you about the research I've been doing the past 2 years on the bioconjugation of anti-SIGLEC H antibodies to bicelles and liposomes for targeted drug delivery of anti-HIV drugs to interferon-producing cells. Whew, all right, that's going to make sense, hopefully, in about an hour. Okay, so roadmap, where are we going today? First, I'm going to tell you about what HIV is and how we plan on treating it, and specifically how we're going to use lipid vehicles to stop HIV reproduction. Then I'm going to tell you about how we can prove that these lipid vehicles are capable of this active targeting of interferon-producing cells, which I'll get into. And finally, I'm going to show you that these lipid vehicles do show good promise as being drug delivery vehicles for antiretroviral drugs to treat HIV. So, what is HIV? Human immunodeficiency virus, or HIV. It's a virus that attacks and infects immune cells in the body. According to the CDC, as of 2021, there's been 1.2 million confirmed cases. And this is a disease where infected cells will release inflammatory chemo and cytokines into the brain, causing inflammation, which ultimately results in negative effects, things like apoptosis, cell death, neuronal degradation, and even patient death. So how do we plan on treating it? Well, as of right now, there's no definitive cure for HIV due to the complicated mechanism by which it reproduces. So we have these antiretroviral drugs that can stop HIV reproduction. So instead of curing the disease and killing all the infected cells, our approach is to mitigate the reproduction of infected cells. This project is going to utilize a combination approach of 3 different drugs that each are going to inhibit a different aspect of HIV reproduction. So to start off, we have dolutegravir, or I'm going to call it DTG from now on, which blocks the active site of HIV integrase, which is step 4 to 5 here, where the viral DNA is being integrated into the cell's genome to reproduce itself. DTG is going to block the site of that enzyme, inhibit HIV replication in that way. Our other 2 drugs, emectrobine, FTC, and tenofovir disoproxil fumarate, TDF, are nucleoside reverse transcriptase inhibitors. That's going to inhibit the step 3 to 4 here, where we're taking the viral RNA from the infected or the infecting cell or the infecting virus and turning it into viral DNA. These 2 drugs are going to stop the enzyme that does that. So by delivering these drugs into a healthy cell that has not yet been infected, we can hopefully prevent that cell from being infected and getting that viral genomic data into its genome so that it doesn't reproduce more HIV viruses. So, how are we going to deliver those drugs? That's what my project focuses on, and I'm going to do it with these things called lipid nanoparticles. So the whole goal of this project is to encapsulate these antiretroviral drugs into a lipid nanoparticle vehicle and attach some molecules on them so that these vehicles can be actively targeted to interferon-producing cells, which are immune cells that are responding to HIV infection that have not yet been infected because they're responding to that HIV infection by releasing interferons to fight that virus. So this schematic here is a bicell. This is a lipid, nanoscale lipid vehicle that we use to protect and deliver hydrophobic drugs to cells. This is a very nice schematic image done by Justin, who could not be here today. He got food poisoning, but this is Justin from the knee lab. He made these very nice renderings from our paper 2 years ago. Does not feel like 2 years ago, but don't go to him with any requests to make figures because he's busy enough as it is. But I'll point out whenever there's a nice figure that's beautifully made by him. But these, what you're looking at here, are called bicelles, which stands for bilayered micelles, which I won't fully explain. But basically, we have a bilayered membrane in a disc sort of conformation. And this is made up of phospholipids. So if you're familiar with cell membranes at all, you know that These are very reminiscent of what a cellular membrane looks like, besides the fact that they're discs and not big, long spheres. So I'll get into how we make these in a minute, but some key things to look at right now before I move on is that these are discoidal shaped, so what that allows us to do is have a greater surface area to volume ratio, which gives us better cellular uptake in the body than something like a sphere would. And they're small enough to be able to cross the blood-brain barrier, which I'll also talk about in a few slides. Another key feature is this long polyethylene glycol tail that comes out of them. This is really where I'm doing a lot of the interesting chemistry. This is where I'm attaching my targeting antibody to my structure so that these things, when I load them with my antiretroviral drugs, they can go to the site of infection or the site of pre-infection to deliver these drugs to stop HIV replication. So we're loading the drugs into this hydrophobic rim region right here where this is a gold nanoparticle, but this is where the drug would be. I'll talk about why the drug would be there maybe a little bit later, but the drug goes into this rim region here, and then we have our targeting antibodies sticking on these purple tails that are out in the aqueous environment, and that allows this whole big system to get targeted to interferon-producing cells, stopping HIV. Okay, so how do we make these things? Well, lucky for us, phospholipids are both hydrophobic and hydrophilic. They have two distinct regions, and this lets them actually self-assemble. Dr. Nee's lab is not actually called Dr. Nee's lab. He didn't want to put his whole name there. He named his lab the self-assembled functional nanoparticle lab. And so we're really interested in these phospholipids that when we put them in an aqueous environment, they self-assemble into a structure that's thermodynamically favorable. But this is not just a single type of lipid that makes up this cool structure. We have many different lipids here. So we have DPPC, which is our main long chain lipid. It has this long hydrophobic region, two acyl chain tails, and then a hydrophilic phosphate head. And that's this blue lipid that you see in this schematic, making up most of this like face region, we call it, of the bicelle. And it forms this kind of planar bilayer, which if we just put DPPC into a solution, it would just make these long sheets forever. We also have DHPC, which is our short chain lipid, which stops DPPC from making that long planar bilayer forever. We want to make discs, right? So we put in this short chain lipid, which is this red rim region that you see here. And this lipid naturally wants to curve, so it, through some thermodynamic magic, will cap off our DPPC regions to make these beautiful discs. These two long chain lipids on the right are our charged lipids. They're very similar to our DPPC, this blue lipid. So they insert along the face of our bicelle, and they give our bicelle a surface charge to enhance stability, because if you imagine two or more of these in a solution, we don't want them fusing together. So we give each particle a like charge so that they repel each other a little bit. And then at the bottom here, we have our two pegylated lipids. Those are our lipids that stick out with these long tails into the aqueous environment. And I have two because one of these is our functional group, which I'm going to talk about in the next slide, that does the cool chemistry where we're binding this targeting antibody to the formulation. And the other one is our control group, exact same thing, exact same length of this long squiggly chain, but it doesn't have that functional group at the end that we care about. Okay, so I talked a little bit about lipids self-assembling, but how do they actually do that? Or why do they actually do that? Well, we have this parameter called the critical packing parameter, or CPP. And it's a function of the volume of the acyl chains in our lipid over the optimal interfacial surface area of the phosphate head times the acyl chain length. So if you imagine a lipid in the bottom here, and this is a figure made by Justin again, you can tell because it's so nice. Starting off with a low critical packing parameter, you can kind of see this is our short chain lipid here who likes to curve. So you can kind of imagine that the phosphate, if we hold the phosphate head kind of constant in size and volume, and we're just looking at the acyl chains. You can see the acyl chain kind of as a function of volume forms this cone shape, where the hydrophobic region is small in comparison to the phosphate head. So what does this thing want to do in solution? It self-assembles into a spherical particle. As we start to increase the critical packing parameter up to values of 1, we start to get less and less curvature as our acyl chain kind of takes up more volume, until we eventually get that planar bilayer conformation, which is what you see with like something like DPPC, where you can kind of imagine if we combine this planar bilayer with a spherical region, we can get those discs that we so love. So I won't go into the minutia of how I form these bicelles exactly, but basically by heating up and cooling down the sample, we can get the short chains to aggregate to cap off these DPPC regions. Okay, so now let's talk about how I can bind this targeting molecule that I've been talking about. So the targeting molecule in question is an antibody. It's an anti-SIGLEC H antibody, which is, SIGLEC H is a lectin. It's a protein that's expressed highly on interferon-producing cells, the cells we want to go to. And by adding an anti-SIGLEC H antibody onto our formulation, we can target the cells we're interested in. So how do we do that chemically? Well, remember I said this PEG group that we're attaching to our bicell has a functional group at the end of it. This is called malamide group. And it's this weird ring-looking structure that does a very simple chemical reaction. I'm not a chemist, so this was perfect for me. I think it was hand-chosen for me, actually, by Dr. Lin in the BME department. The reaction involves a sulfhydryl compound, which is abundantly present in most proteins, especially in antibodies. It holds together the two heavy chains of antibodies. And a maleimide group. In aqueous solution, this reaction just proceeds forward spontaneously. So we're able to attach any of these free sulfhydryl groups to our antibody via a double bond attacking this. So this is the reaction simplified here. And you can imagine this is our functional group. It is attacking that sulfhydryl compound on the protein and giving us a stable bond. Cool. So now we have our antibody being attached onto our bicellular formulation, right? How do we prove that that's actually happening? Well, it's been well reported in the literature that this reaction happens with a variety of protein, pretty much any protein. This maleimide can do this conjugation too, but we wanted to prove that our whole lipid system, this whole bicellular system, can attach to an antibody. So we started off, this is the very first test that I did, trying to determine, does our bicell formulation link up with that, with an antibody? Does it do that as a whole system? So the procedure here is I loaded my bicelles with a Nile red dye so we can visualize them fluorescently. And I have this antibody. It's not our anti-SIGLEC H antibody of interest. It's just a basic Alexaflor 488 secondary antibody, but it has those sulfhydryl groups in it. So we can do this maleimide binding. So the procedure here, what you're looking at is the first two columns are just channels alone. So the first columns are green channel, our Alexaflor 488, which is our antibody. And the second column is our Nile red signal from our lipids. And so the procedure here was to dot on just a little bit of solution of the antibody, rinse it off any unconjugated things, any unconjugated antibodies that weren't stuck to the slide, and then add our lipid system, wash away anything that's not conjugated, and see if there's co-localization. Basically, are these two things showing up in the same area? That would tell us, okay, maybe even if there's a non-specific interaction happening here, do our maleimide lipids, do they co-localize with antibodies in general? And so these rows that you're seeing here, the different numbers, these are different charge ratios. So remember I said we have those charged lipids that promote stability. I was testing a variety of them, some positively charged formulations, some negative and some very negative. That's all that you're looking at there. And so these images are from our maleimide containing bicelles. So all of these should show good colocalization. And it's kind of hard to see, but there are yellow regions which show overlap between green and red signal. And I'll get into the quantitative data in a little bit, but this is, these are formulations that should be, according to what we've seen, should be colocalizing with our antibody. These are the control samples that shouldn't be colocalizing with our antibody. So these don't contain maleimide group, just that control peg. And these are the images from that same format. And here's the data quantitatively laid out via a colocalization analysis in ImageJ. So here we're looking at the z-score of colocalization. Not exactly in these super high intensity regions, because that's kind of a grouping. You can kind of imagine it as precipitated antibodies. We're looking more at this diffuse staining region here, and in between you can't see it because it's not super blown up in contrast, but there is signal coming from these inter regions between the super bright signal. So we're looking at the colocalization correlation within that area, within those areas. And what we see here in all cases, in all different charge ratios, the blue is the +5, green is -5, orange is -10 charge ratio. In all formulations, the maleimide group is significantly higher in correlation to our control group. Basically, our maleimide formulations can colocalize to our antibodies in a much higher, in a much greater way than our controls do. So this is great. Our formulations look like they're colocalizing awesomely. And so, next step, we wanted to start moving into in vivo after this, or in vitro at least. But before we did that, our collaborator who originally came to us with the proposal for this project wanted to just verify that these formulations can pass the blood-brain barrier. That's an important step for this project as a whole. So over at the Texas, University of Texas, Rio Grande, they have an artificial blood-brain barrier set up. And they can perform analysis on it to basically measure the concentration of whatever we're interested in. In this case, it's our bicelles and Nile red dye, which is that red dye, hydrophobic red dye that we encapsulated in the bicelles. They have the system for measuring how much these components pass through the blood-brain barrier over the course of 2 hours. And so we sent our samples over to them, and they did verify that, yes, we do see that the concentration increases over the course of 2 hours. Basically, these formulations are capable of passing the blood-brain barrier, which is what we expected. That was great. Then we ran into our first really major issue. We wanted to start encapsulating these antiretroviral drugs, right? We're at the point where we've shown that this maleimide reaction can happen, and now we want to put our drugs in and see if we can, get these drugs delivered to interferon-producing cells. Well, here's the issue. If you remember back to the structure of what a bicell looks like, the only way that we can encapsulate something into it is if it's hydrophobic, right? Because we have that hydrophobic region, and we need a hydrophobic drug that's going to want to spontaneously kind of be in that hydrophobic region. So that's great if we're encapsulating a drug like DTG, which is water insoluble. And this, what you're looking at here, is a hydrophobicity plot made by the website Mole Inspiration. When you give it a chemical structure, it'll break down how hydrophobic or how hydrophilic different regions of the drug are. And so hydrophilic is very red. As we go more hydrophobic, it goes into the blues. DTG, as reported by the literature, water insoluble. So this is a great molecule if we wanted to encapsulate this into a bicell and deliver it to interferon-producing cells. The issue here is our other two drugs. FTC and TDF are both water soluble and not very hydrophobic, so these are not going to naturally want to go inside, be encapsulated by our bicelles. And this issue first presented itself when we realized that when we were forming our bicelles, our solution wasn't transparent like it normally is. It's opaque, meaning we don't really have bicelles anymore, because as a feature of how small bicelles are, the solution is just going to look transparent because all the light is transmitting straight through it. There's no scattered light. Liposomes, however, which I'll show in the next slide, are big enough to scatter light, so you start to see an opaque sample when you look at that. When we were trying to encapsulate these drugs into our bicelles, we were getting opaque samples, which indicated to us, okay, whatever structures we have in our samples, they're big enough to scatter light. So we're looking probably at liposomes. And when you measure them, that's exactly what we saw. We were seeing liposomes, and so at this point, the project is pivoting to go with those liposomes. If the structure is going to naturally form liposomes when we add these drugs, might as well just hand tailor the project to use liposomes to deliver these drugs, right? So this is what a liposome looks like. You can kind of imagine it as well, this image isn't like big enough to imagine it really, but you can kind of imagine a liposome as a bunch of bicelles fused together. Each of these regions could be like a bicell, and then you add another bicell rim to rim, and you would eventually get this liposome. And that's what we think was happening when we were adding these drugs. So in the sake of saving material, because these drugs are very expensive, we wanted to try to basically form these liposomes from our bicelles via this procedure of heating and diluting our samples, which is basically forcing our bicelles to fuse together to form these liposomes. And when we did this procedure, what you see in this graph that you're looking at here is over the course of 72 hours of heating up these samples, how does the size, the radius change over the course of time? And so what we see here in all cases is the size is increasing because we're allowing these bicelles to fuse and fuse and fuse into these liposomes. But the size is super uncontrolled. We have a huge variety here in size. 300 nanometers is gigantic. Probably not going to get any valid cellular uptake with a liposome that's 300 nanometers in size. It's not going to be able to cross the blood-brain barrier with that size. We have some that are less than 100 nanometers. And this is not very conducive for the next experiment I want to run, which is measuring the conjugation of this targeting antibody via a discrete change in size. So we move to a new method, extrusion. So instead of forming these liposomes by heating up and diluting our sample, we turn to extrusion, which is basically where we're passing our lipids back and forth through a filter of a certain size to get a very homogeneous sized liposome. And so this is our instrument for that, just 2 syringes, filter in the middle. And you can see initial particle size distribution. The x-axis here is showing our size, starts off at about 1,000 nanometers, and over the course of 11 passes, our size distribution shifts down to 100 nanometers. One big peak shows us that all of the particles of interest are at 100 nanometers in size, which is perfect. This is exactly what we want. This isn't my data, though. This is just representative data. Let's go to my data. So these are our formed liposomes, size measured, post-extrusion, and with antibody. So the idea here is that if we have a really well-controlled size, when we add our antibody, we should be able to see a really marked size increase when we add our antibodies, demonstrating that our antibody is binding to our liposome. And so PG is referring to our negatively controlled liposomes in this case, and TAP is referring to our positively charged liposomes. And then we have our controls and our malleamides, which should be binding our antibody, right? And so here's the data. PG control does exactly what we expect. There's no change when we add this antibody in size. Great. The PG with our malleamide group, when we add our antibodies, we see this really marked size increase of, I think it was exactly 7.5 nanometers, which is what we expect for an antibody size change. So this did exactly what we expected it to do. And now TAP is where it gets a little interesting. These are positively charged liposomes, and they both, when we add our antibody, both the control and the malleamide formulations decrease in size, which is really strange. We don't expect this to happen much. with anything when we're measuring size, really. But it does happen. So what's the explanation for that? Well, I'll tell you about one current hypothesis that we have for why this size is decreasing. And it's related to the polyethylene glycol, these pegtails. My formulation has a lower than 5 molar percent peg making up this liposome. So what this pegtail kind of adopts as a conformation is this mushroom shape, where it doesn't really stick out straight like this brush shape on the right here. It kind of coils up along the surface. And so as you increase in concentration, you get more of this peg kind of sticking out of our structure. So this can kind of give us some insight into why our TAP samples are decreasing in size. Because if you imagine, TAP is surface charged with a positive characteristic, right? So that's this light blue area, that's where the surface charge is. If this is positively charged, and we're attaching a negatively charged antibody to the end of these pegs, there might be some ionic interaction happening here, where when the peg attaches the antibody, it kind of starts to want to lie flat down. It has this room, this steric ability almost, where it can lie down on the surface of the liposome and kind of decrease its size. If you imagine in the wild, animals will like puff up their fur when they want to look bigger. That's what is the opposite is kind of happening here, where instead of puffing up and looking big, when we bind this antibody, the fur or the peg kind of lies flat down, and that could be resulting in this size decrease. There's also kind of not evidence, but it kind of makes sense when you think about TAP as our positively controlled or positively charged formulation. Our peg lipid actually does have a little bit of a negative charge at the very end. So even though we're at less than 5 molar percent in our composition, our peg in our positively controlled samples could be looking more like a brush-shaped peg just due to this repulsion from the surface charge of the liposome in general. So we could be looking like this brush shape initially. That's why the TAP may be so much higher than both PG samples initially. And then when we add our antibody lying flat on the surface, that could be what's resulting in this size drop. There's other ways that we could investigate this phenomenon. We can look at some electron microscopy data to see if we see any interaction happening there. There's different forms of light scattering that give us size, but not exactly the same size. We're looking at hydrodynamic radius here. If we look at something like radius of gyration, that would give us a little bit more information about the size of our particle and what's actually happening when we add this antibody. But this is what we have for now, and overall we still think it shows good evidence that our antibody can be bound to our lipid system in general. Okay, so we have our system pretty much fully ironed out. Now we want to start getting to the point where we want to make sure that this targeting mechanism is doing what it should be doing, which is targeting these interferon-producing cells. So now we're actually in a brain section. Before with the microscopy images, it was just on a slide as solution, basically. Now we're actually in mouse coronal brain sections. And so you'll notice there's no green signal here. That's because we don't have a fluorescent antibody. We just have our lipids loaded with Nile red dye. And so in this case, we are actually using our antibody of interest, this Anti-SIGLEC H antibody. And so we should be expecting some sort of differential staining pattern that shows us our lipids are accumulating in the area where interferon-producing cells are at their highest. And I don't have a marker for interferon-producing cells here, but we do have evidence from the literature that along this border region of brain tissue sections, there should be higher density of immune cells in general, right? It's the blood-brain barrier. There should be a lot of immune cells protecting our brain from pathogens. So we should be expecting high density of interferon-producing cells along this border region. So the top row here, what you're looking at is formulations that were incubated with brain sections that contained this targeting moiety, this antibody. On the bottom row is the exact same formulations incubated with the brain section, but without this targeting antibody. And so you can kind of see with your naked eye here, and I'm going to get into the quantitative data, but you can kind of see that in these top images, this border region, shows a much higher staining intensity than the internal area, what I call the body. And that's not the case in this bottom row, except for this first image a little bit. We have pretty much diffuse intensity from the whole section. And that's also demonstrated by what you're looking at up here, these plots, which I'm going to blow up in the next slide. But these are plots of intensity over a spatial area. So what I did was draw a line perpendicular to the brain section, starting from outside, going through the border and into the body, and we see the intensity as we trace through that distance, right? And so we start off super low when we're on the outside of our section, we get this big peak when we're at the border, and then in our first row here, what happens is after we pass that border region, our intensity drops down in all cases, but when we're looking at our bottom row here, our intensity jumps up when we hit that border region, and then it stays pretty consistent after that. So we're looking at some sort of differential staining pattern here. Here's those images again blown up. This is the top row, jump up and then come back down, jump up, down, and here up, a little bit down, but up and stable for the most part. And so here's that data all averaged out and put into a nice statistically significant bar graph. We're looking at the average, on the y-axis, the average difference between the border and the body in intensity. So border region subtracted from the body region, how much more intense is it in our antibody-containing formulation incubations versus our non-antibody containing ones? And we get a very significant difference there in intensity in our border regions with our targeting antibody. Great. Our system is doing exactly what it should be doing, and it's targeting where we think at least these interferon producing cells reside. Awesome. Went faster than I expected. So, great. We've shown that our system can bind with our targeting antibodies in exactly the way we expect. We showed that there's a differential staining pattern here, so they should be doing that targeting that we expect them to do. And now this is all that's left. We want to put the final nail in the coffin with showing that our system can bind these targeting antibodies by doing some sort of spectroscopy-based binding assay to show, okay, if you know exactly how much antibody you put in your preparation, you should be able to determine exactly how much protein is in those samples, basically how much of our antibody is being bound by our lipids. So that would be a nice metric to have. How efficient is our system at binding our targeting antibody? Also, another next step is optimizing this vehicle, this liposome vehicle. There's a lot of things you can change about it. Namely, surface charge is a pretty easy thing to change. And as you saw before with that size differential when we were looking at dynamic light scattering data, that was quite variable depending on the charge of the formulation. So altering that could give our vehicle potentially better outcomes when we start going into the patients as far as drug uptake, and I can get into that as far as how the charge impacts cellular uptake. You can also change the composition of the actual lipids. So I was working with those DPPC lipids, which like to be in that planar region, which is great for bicelles, not exactly perfect for liposomes where we want a decent curvature. So putting in a different lipid to compose most of the liposomes could be helpful in that case. And finally, moving on to in vivo testing to show, does our formulation with all of our encapsulated drugs, does it do what it should do and stop HIV reproduction? Okay, and that is all. And so many people to thank. Almost got everyone in this room. Justin, who again made a lot of those early figures, couldn't be here. He had food poisoning from the knee lab. Dr. Chung Hao, who has now since graduated and is working at Oak Ridge National Lab. He was a fantastic help my first couple years as an undergrad especially, but also my first year as a graduate student. Everyone else in the knee lab, a bunch of which are here. The Laturco lab, Jorge, who couldn't be here, great friend, great member of my cohort. Kevin, who showed me a lot of the ropes in the lab, as well as the two undergraduates, Jackson and Vadeep, showed me so much as undergraduates. I was the graduate student coming in, probably should have known more than them, but gosh, were they helpful in knowing all things microscopy and P&B related. I was coming from the BME background, sort of. My committee members, thank you all for being here. Collaborators, Dr. Upal over in Texas, who came to us with this proposal. My P&B TAs, Will and Andre, helped me through those those early P&B courses when I was an undergrad here. Appreciate that. Rick, great friend. And of course, my admissions committee, who actually let me get into this program in the first place. And last but certainly not least, my future wife, Lily. So supportive, really the best teammate I could ask for these past 5 years. And that's all. Thank you, everyone.
For questions?
Lots of time for questions. Duran.
So, I mean, I've seen you around the lab doing a lot of those extrusions.
Yes.
Manually, but I've always been curious, why not use like a syringe pump? Like, is there a specific reason that you need to do that manually? Is there a feel to it, or something that you could like kind of program?
So there's two things. First of all, for this extrusion to take place, we need to be at a temperature above the melting temperature of our main lipid, in this case DPPC, which is above 41 degrees Celsius. So we have this whole thing on a hot plate, which kind of complicates things spatially, I guess, to put a syringe pump there. If you had some sort of specialized syringe pump that like had its arms out, you could maybe do something there. If you like stabilized the whole system and locked everything down. You do kind of run into an issue though, if your pump is too strong. What we see sometimes, I've never seen this, but Justin certainly has, and it's a big heartache. Your filter can break if you press too hard. So that can happen if we're using a syringe pump. Your filter breaks, and then when you finally get your solution out, so sometimes your solution will just leak out of this whole apparatus. But if you do get some solution, even if your filter broke, you'll probably get something like this, where you have two different size distributions, and that's just no good. Rick.
First of all, great talk.
Thank you.
Your talk inspired me. I want more BME and PMB crossover.
Yes, BME P&B crossover is the best.
Especially like a procedure or treatment that can cross the blood-brain barrier is really impressive.
Yeah.
But actually, on your grade values, when you were determining that on the blood-brain barrier, on the border,
Oh.
I'm wondering if there's ever been, or if you've noticed, or if there's ever been any measurements for diffusion. Like, what is your time scale for actually identifying it on the blood-brain barrier border? Meaning, if it's going to bind and it's going to be there, is there a timeline in which it does sequester that?
I see what you're saying. Ooh. Not that I know of, but that would be an interesting test, definitely, to do how fast these are accumulating in the blood-brain barrier and how quickly they're aggregating there. That'd be, I expect, pretty much instant. Once the sample reaches that area, the antibody is going to see its epitope and it's just going to lock on. So quick, fast. It's going to happen fast. Does that answer your question? Yeah. Okay. Duran.
For that in vivo testing, are you going to go kind of, would you proceed with like right into organoids, rather than into mice, or?
So the antibody that we're interested in, it's an anti-human SIGLEC H antibody. So we would have to do it in a human spheroid, if you have any spheroids that you want to cook up for me. But we could definitely start in mouse using some sort of equivalent. Either of those work. Yeah, either. Mike and then Will.
So the liposomes are larger than the bicelles?
Correct.
Are they still small enough to cross the blood-brain barrier?
Yeah, that's a good question. They can be. So in HIV, what we see is the blood-brain barrier does get disrupted. as with a lot of these brain-related disorders. So what we have is something called the EPR effect, this enhanced permeability and retention effect. I'm going to go back to like my first slide. This doesn't really show the blood-brain barrier, but essentially what happens in these disease states is because we have so much inflammation, the gaps that are normally really small and hard to get through in the brain start to kind of increase a little bit, and we can pass through larger particles. So it does present a little bit of an issue as far as being around 100 nanometers in size. Definitely not the most ideal case for passing the blood-brain barrier, but if we're working in a disease state like this, there is some hope of getting this delivered into brain cells. Yeah. Will.
So this is kind of like two wrapped up into one.
Okay.
What actually is SIGLEC H, if I'm saying that right?
Yeah, yeah.
And then what? How specific is it to these particular cells?
The interferon-producing cells.
It's not specific. Is there any sort of like worry like off-target effects?
Very specific. So SIGLEC H is an immunoglobulin lectin protein that binds to carbohydrates in these interferon-producing cells like macrophages. And it's really only expressed in these interferon-producing cells, so exactly what we want to target. That being said, even if we are having some non-specific targeting with, if there was a SIGLEC H expressed on a non-interferon-producing cells, we're still accumulating mostly in this area that we want to be, in this high immune cell area. And if there's a little bit of bleed over where our active targeting maybe isn't as specific as we want it to be, we can do more. We can add more targeting moieties in exactly the same way and have even more specificity as to where we're targeting. And that would solve that issue. But overall, SIGLEC H for interferon-producing cells, pretty specific. Any other questions? All set? Okay. Thank you all for coming. Okay. I don't know what happens now. Yeah, you guys. Do I stay here and you guys ask me a bunch of questions?
Yeah.
Awesome.
By the way, just quickly, thanks to all the students who came and supported. Like, it's the middle of a Wednesday. This is really
Yeah, thank you so much.
It's really cool you did that. Thank you.
Thank you.
Good to see you.
Always got the good questions.
Of course. Well done, Luke. Hopefully I'll see you in lab.
Thank you. Yes, maybe. Yeah. Yes.
I'll keep you updated.
Thank you.
See you guys.
Do you remember him?
I do. I'll tell you, there's a weird cognitive thing. So I don't think of you as an undergraduate anymore.
Okay.
I realize he's so evil with you, EVA. I mean, he's the same age.
Mm-hmm.
But because I only encountered him as an undergraduate.
Ah, you got that link.
Same age.
That's interesting.
Not